before you became a scientist here, what education did you have prior?
Well, I went to the graduate school, first go to the master program, then PhD program, and I did my master thesis work in California and working on the melanoma research. And I went to the University of Illinois Urbana-Champaign. My major is immunology, so I studied the antibody to treat the cocaine addiction. So the biomedical research program actually is the most interesting to me. And I became more and more interested, and I did more in my postdoc. And so it's actually has to take 3 years master, 5 years PhD, and 5 years postdocs. That long time to get into the field. And I also work for a very short period of time in industry, like a biopharmaceutical company in California. And I was very excited about the drug development. So like what I'm doing now is more focused on the drug development for cancer therapy.
Okay. How does your job connect with biochemistry?
Well, biochemistry is the foundation for almost every biological research. And I actually teach biochemistry in NIH since 2006, and the graduate program, and that's the whole year course. And I found that biochemistry, the basics in the biochemistry are very important for any researchers in the biological lab or cancer lab or neuroscience lab, or the other buildings, neuroscience centers. So regardless what area you are working on, biochemistry is the foundation for researchers to understand the mechanism underlying the biological pathway or process, and to be able to disrupt it if you want. For example, in the case of cancer, you want to disrupt cancer cell growth.
So my project is about biochemistry. The topic I'm researching is biochemistry.
Okay, yeah.
And so, what tools do you use in your lab?
Well, we are using common biochemistry tools such as SDS page, Western blot, protein purification. We use all kinds of tools to purify protein, such as ion exchange column, affinity columns, and size columns. So based on the property of the proteins we work with, we purify protein based on the charge, based on the size, based on the tag attached to the protein, so we could use affinity columns. And we also do PCR, molecular biology, and we do the sequencing, and we do cell biology, microscopic confocal staining, immunostaining. So those are common technologies. So one thing we are very unique, our lab is quite unique, is I don't think everyone in any lab could easily do it. We actually do a technology called phage display technology. So it's a bacterial virus, and display the antibodies on the bacterial virus. And we made it as a library, and use the library to screen the cancer drugs. And that technology is a biochemical technology again. It has been around for more than, I would say, almost 20 years. But since the technology requires very sophisticated biochemical skills and a very in-depth understanding of the procedure, and also need exceptional patience, because you cannot see it, because it's a virus, it's a bacterial virus, you cannot see it. So you grow them in the E. coli, but you really cannot see the virus, but you have to isolate the antibody, based on how they bind to the target. In our case, it's cancer cells, and so that's why the phage display technology you just see in the hallway. So we have just published a paper in PNS 2 weeks ago, and that study is actually used the phage display technology, and it's quite unique. Even very well trained biochemist, not everybody could do it. It's just a very complicated technology.
Also, what skills do the practitioners in this field use?
So the skills, regular skills is you need to understand the proteins. So that means you have to run the protein on the gel to understand the size of the protein, the charge of the protein, and modification of protein, whether they have sugar or not sugar, if they have lipid or not have lipid. So those things, the basic properties of a protein. On the other hand, you need to learn the molecular biology. So you need to learn how to clone the gene. You need to express the gene in mammalian cells or E. coli. Some people do actually express them in yeast. So that's kind of basic skills you need in the lab. And also, as you see, the microscope, you need to learn how to examine the cells under microscope to determine whether they are healthy, whether they are differentiated very well, or they are growing as healthy as you expect.
Okay. And so do you have a role model or a person that you greatly admire in this field?
Well, I think there are many role models I will imagine, but since you asked this question specifically, I would think one of the Asian American in the NI. Actually, I think maybe 2 Asian American before me working in NIH as a scientist are very good role models, and one is Dr. Lee. I could show you after interview the portrait there in the building 10. So he worked in a clinical center. So I could show you the picture. You could take a picture there. So I think it would be nice to see his picture. And he worked in late 1960s and 1970s, before most people even were born actually at that time. And he originally came from China, and he worked in NIH as a scientist. But at that time, it's just past soon after the U.S. Terminated the law actually, which is kind of against the Chinese American in this country. So in general, people still kind of probably not fully appreciate the contribution of Chinese American to this society. But at that time, he was a scientist here in NIH, and it was very few, of course. And he worked in the Building 10 as a clinical scientist, and he ran the first chemotherapy in the world to treat cancers. The cancer he treated is a women cancer. I believe is a very rare women cancer. It's like a cholangiocarcinoma. So this cancer is very deadly cancer because 98% of the women actually died of this if they got this cancer. And he treated with a very high dose of chemo drugs in late 1980s, late 1960s. And after treatment, I believe at least more than half of the patients he treated, they recovered. So this is the first time in the world and in the history, and a scientist shows chemotherapy actually could treat cancers. Before, people just think chemotherapy could be a kind of supplemental, not the real treatment. It's just the surgery is the major treatment. Chemotherapy just as a supplement And he actually shows, okay, chemotherapy could really treat cancer. And you see what we are doing now, what my lab is doing, like antibody therapy, is kind of chemotherapy also. So he is the pioneer, and he is a Chinese American, and he did the first. But at that time, and he was not fully appreciated. I think mostly because he was Chinese. Okay, so he was actually fired by the NIH and by his supervisor or someone else. And he finally found a job in New York, then moved to California. I believe he finally died in San Diego. And fortunately, before he died, and he got the Lasker Award. That Lasker Award is like considered as American Nobel Prize, and it's for clinical the greatest achievement for someone who has such a great clinical contribution to the mankind. And of course, he and ironically, he and his supervisor in NIH, and they both shared the Lasker Award. And of course, his contribution is fully appreciated. So now you could see his portrait in NIH actually, and we actually memorize him as a hero. Yeah. But it came for a long way to be Chinese American scientist to be fully appreciated in this society.